everybody. My name is Phillip Richmond, and today I'm going to talk to you about the multi-omic approach to identify markers of cerebral demyelination in X-linked adrenoleukodystrophy. I've just added this doctor to the front of my name as I finished my PhD last month at the University of British Columbia with my research happening at the BC Children's Hospital Research Institute here in beautiful Vancouver. So phenotypic discordance in XALD is a very widely known phenomenon. And a particular aspect of that is the development of cerebral demyelination. So throughout the life of a patient, it's possible, because of some unknown factors, or what we'll call modifiers, essentially the development of cerebral demyelination occurs. And this happens in about 50 to 60% of cases, and it causes blood-brain barrier disruption and increased inflammation. Now, the diagnosis for cerebral demyelination is done by recurrent MRI and intense psychological evaluations, and it's very important to catch early because if left untreated, it can progress to a vegetative state and death within 2 to 3 years. There is a treatment option, which is to use stem cell therapy, which does rescue the continued demyelination phenotype. Now, there have been several genetic investigations to try and find modifiers demyelination in XALD. Now, a genome-wide association study where we essentially look at all of the candidate genetic loci has been limited because large sample sizes are needed and they're very challenging to acquire, especially for a rare disease such as adrenoleukodystrophy. Candidate gene association studies have been done, and they are limited because essentially what happens is there's very limited replication of the different genotypes which are identified. Typically what happens in a candidate gene study is a gene within a targeted pathway, for example, the fatty acid metabolism, is queried for polymorphic genetic variants between cerebral demyelination and no cerebral demyelination. And there are some candidate genes such as CYP4F2 and ELO-VL1, which do show some signal that could potentially indicate a predictive of cerebral demyelination phenotype. Beyond genetic investigations, there have also been recent what we will call alternative profiling. So profiling the proteins or profiling the DNA methylation and gene expression in both plasma and cerebrospinal fluid. Now, these two papers, this one in 2019 and this one in 2018, did look across patients that had developed CALD and saw, in one case, an overabundance of a protein called profilin-1 in both the plasma and the CSF. And then in the other paper, several genes with implications in altered DNA methylation or gene expression, which could be capturing measurement of the environment or the state of the cells. So the problem that we're faced with is that even in spite of these previous results, these results are indicating that there is some portion of CALD patients which may show this phenotype, and the same for essentially the genetic and the DNA methylation studies. So to date, it is unclear as to whether there is a single marker which can predict the development of cerebral demyelination in these patients. Now, it's very beneficial if we can identify such a marker, as it could be used to replace the current protocol of MRI scans and intense psychological evaluations for diagnosis or prognosis. So what if we could replace all of that with the blood test? Which leads to our research question, can we identify a molecular modifier signal from a blood sample which is predictive of the onset of cerebral demyelination? Now, our approach to this was to look pretty much everywhere that we could. So what we did was we first started with 6 brother pairs where they share the exact same ABCD1 mutation, but one brother will have not developed CALD and the other will have developed CALD. Now, for all 12 of these individuals, or these 6 brother pairs, we performed whole genome sequencing, DNA methylation profiling, RNA sequencing, and LCMS for proteomics and metabolomics analysis. So from the blood, we isolated either lymphocytes or plasma, essentially ran all of these different assays, processed the data, and performed group-wise comparisons where we compare all of the CALD patients across all of the non-CALD patients. Now, multi-omics projects produce a lot of data. And if the talk was a full hour, you would see all of that data. Unfortunately, I will just be able to provide you some highlights, and hopefully we can have some time for discussion at the end if anyone wants to see some of these slides. So as a summary of the results, unfortunately, we did not find a confident single differentiating signal across all brother pairs in any of the omics datasets. Now, when we compared the DNA, we find, first of all, no evidence to support previously proposed modifier alleles. And we did find 2 genetic variants, although with low predicted impact, which can discriminate between all brother pairs. In the transcriptomics, proteomics, and DNA methylation data, we do not find a signal which separates all 6 brother pairs. However, we do recapitulate the signal of PFN1 overabundance in a subset of CALD patients. And we also observe a gene PTPRN2, which had differential expression and methylation in a subset of patients. In the metabolomics data, we saw clear differentiation between ALD and normal patients. However, or ALD and normal controls. However, we do not see any difference comparing cerebral ALD versus non-cerebral ALD. And intersecting these different omics data sets did not provide us any additional statistical power. So what are the key takeaways from this study? It was a large study, which we believe, based on this data, that there is no strong single predictor of the onset of CALD, which can be taken from blood. However, it is possible that some signals have predictive power in a subset of patients. So the onset of cerebral demyelination is not controlled by a single locus and is not measured by a single molecular signal, but perhaps a combination of them is still possible. So summarizing our hunt for modifiers, our strengths of our study, the lipid profiling confirmed the known profile for ALD, although not much similar between, or there were not many differences between CALD and non-CALD. Discordant genetic variants, which we did identify, would be interesting to confirm in a larger cohort to see if these are just discordant genetic alleles based on the fact that we expect them by random, or whether there's truly a functional component to those discordant genetic alleles. We also believe that the data for these brother pairs can be used in future studies by aggregating our analysis and our data set with future larger data sets, or to confirm other hypotheses generated from smaller studies. We also found it quite interesting that we confirmed PFN1 overabundance in 4 out of 6 patients, and it could be interesting to confirm in a larger cohort. Now, the limitations and pitfalls. We had too few individuals to have any confidence in multivariate differences between phenotype groups. So if we did want to consider multiple different genetic loci, or if we did want to consider multiple different molecular markers, we are too limited with only 12 samples. Furthermore, there's a chance that patients can develop CALD throughout their lifetime, especially in early adulthood, and this lowers the confidence in our genomic discordant results, as some of our patients were only in their teenage years. We also see that there's a high amount of interfamily variability in these multi-omics assays. Now, additional time points or additional samples from the same individual can help account for some of this variability and noise in such massive data sets. So what are some recommendations for future studies and some next steps? Well, first and foremost, we need to assemble larger cohorts. Patient participation is key as the data can be noisy, and the statistical analysis is limited when you have few data points. For genetic studies, we need to identify discordant brothers who are later in life and therefore have a lower chance of both brothers developing CALD. If we can take multiple samples per individual, this will also be ideal. Another measurement of multi-omics, which can capture the role of the environment, is the microbiome, which has been implicated in several different human phenotypes and would be interesting to examine in this context. Our next steps are to assemble a larger patient cohort to aid in the statistics and then validate some of our findings in such a larger cohort. This paper can be found here in Frontiers in Cell and Developmental Biology. And we also just published a commentary this week discussing the findings and future directions for both ALD studies and other modifier diseases. I'd like to acknowledge the project lead, the multiple team members, both in Amsterdam and here in Vancouver, the adrenoleukodystrophy cohort from Amsterdam, very much appreciative of patients and their families, and funding for myself. And I see that there are some questions in the chat, and I have just about a few minutes to address these. Okay. So the first question is, when you say MRI and intense psychological evaluation, what do you mean by the latter? Only familiar with MRI for CALD. Okay, so this is just something that I am not an ALD expert, and this is something that I pulled from some other experts. So I'm not equipped to address that question. Perhaps MRI is really the only better assay to describe there. What other factors were considered beyond multi-omics? There's hypothesis that trauma may set off the cascade. Yes, so this is obviously a limitation in our analysis. All we have is a single time point, and obviously throughout a patient's life, there's a lot more metadata which can be considered. So things like trauma or treatment with specific antibiotics even, there's a lot of things that we did not account for within our study. I have a question in the chat which says, does knowledge of environmental modifiers such as head trauma help search for susceptibility genes? Larger cohorts without attention to environmental triggers may not help. What new methodologies could help here? So more detailed patient profiling, and if there was knowledge such as head trauma categorized and labeled for all the patients in a cohort, that would be incredibly valuable. Now, I'm not aware of such a head trauma labeled cohort, but I will say that it is still possible that head trauma could essentially produce a cascade of molecular downstream signals. And in our approach, we were really trying to capture evidence of those signals in the data. So it's possible that head trauma might create a cascade of perhaps protein signals, which we could then capture as a signal within our data. But I'm not familiar with any head trauma samples so far. I have a question here. Speaking privately. Okay, I'm not going to read that one out loud. I have 1 minute left. Any last questions? Okay, I am asked to direct you to the ALD Connect website to get to the next talk. I would like to thank the organizers for the ability to present here. Yes, aldconnect.org, and very excited to work with such an engaged group of patients and clinicians and researchers. Have a good Friday.
you.